Multiplex Automated Genome Engineering, or MAGE, was created by George Church and researchers at the Wyss Institute as a way to rapidly edit multiple genes across an organism's genome. Prior to MAGE, the process of genomic engineering involved introducing changes to a single gene, introducing that change to a population of cells, selecting for transformants, and then repeating the process for each change you wished to implement. With MAGE, it is possible to generate multiple changes simultaneously across the genome. These can be mismatches, insertions, or deletions. To do this, oligos are synthesized that are complementary to the target gene on the sides and contain the desired change in the middle. The change can be a single base or up to 60 bases, with a total oligo size of 90 base pairs. These oligos are then introduced to the cell using electroporation. The E. coli cell used lacks mismatch repair and can express a bacteriophage-derived single-stranded DNA binding protein called beta protein. During DNA replication, the oligo is stabilized by the beta proteins and attaches to the lagging strand of the section of DNA it is complementary to. A mismatch occurs where the change is to be implemented, but is incorporated due to the lack of mismatch repair. This creates two copies of DNA, one with the desired mutation and one without, which is subsequently sorted into two daughter cells, one with the original DNA and one that has the mutation in one of the two strands of its DNA. The mutated cell divides, creating one cell with the unaltered genome and another with the complete mutation incorporated into its genome. The previously unaltered cell divides to create two cells with unaltered genomes. Copies of the oligo are then introduced to these cells. After 6 to 8 cycles, the mutation is present in 99% of the population. This process does not occur at just one site in the genome. MAGE can target many different sites simultaneously and even target one site with a range of different oligos to generate huge genomic diversity. The process is also automated within the MAGE device. First, the E. coli cells are incubated in the growth chamber until they reach a population of 1 billion cells. This then triggers the next phase, where the temperature is increased to 42 degrees to induce beta protein expression. The cells are then moved to the cooling chamber for cooling at 4 degrees Celsius. This halts cell growth and potentially toxic overexpression of beta proteins. The cells are then moved to the filtering area, where the growth media is filtered off and the cells washed with water. The oligos are then added in the oligo area and then introduced to the cells via electroporation. The cells are then moved back to the growth chamber to repeat the process. This process was first used by George Church to target 24 genes in the DXP pathway to produce E. coli cells with a 5-fold increase in lycopene production over the course of 3 days. And that is MAGE in a nutshell. I hope you found this video both interesting and informative. Thank you for watching.